Hi everyone. In this lesson, we're going to go over translation, which is the process of actually converting the mRNA sequence into a final amino acid sequence, which is basically the protein. So in this case, translation occurs in the cytoplasm of both a bacteria and a eukaryote. Okay. This occurs once the mRNA gets to get into cytoplasm where the ribosomes are located. And ribosomes are the site of translation. All right, and then translation is the actual process of creating the protein from the mRNA sequence. All right, so the genetic code is ultimately the sequence of bases and the DNA and thus the mRNA and how they relate specifically to the outcome of the protein. So in this case, there are 20 amino acids, but there are only 4 nucleotide bases in DNA. And this is sort of a recap of what occurred in the last lesson, but just a reminder, the number of nucleotides that correspond to amino acids are 3. It's the triplet code or the codon. So every 3 bases of the mRNA sequence is ultimately going to lead to a specific amino acid outcome. Now, the template DNA strand and the resulting mRNA really depend on the 5' to 3' directionality. So it's really important that you pay attention. The mRNA is always 5' to 3' in terms of the direction. So whether or not you're going to use a complementary sequence from the DNA to get the mRNA or not is going to depend on the orientation of the DNA. So if we give you the DNA template strand, the DNA that is 3' to 5' that is used to pair to form the 5' to 3' mRNA, then you're just going to do the complementary sequence. U is complementary to A, G is complementary to C, the C is complementary to G on the mRNA strand, etc. But if we give you the 5' to 3' DNA strand, be careful, because if you just give the complementary sequence, you're actually not going to give me the correct mRNA sequence. So do pay attention. I'll give you some examples of those problems towards the end of this lecture. Okay, another thing to consider is why do we use 3 letters rather than 2 letters or 4 letters? Well, you need to remember that the number of amino acids that we need to code for is 20. There are 20 unique amino acids available to help create proteins. Okay, so we need just enough base pair combos that will lead to at least 20 different unique combinations. If I did just 2 letters combos, well, if you multiply the amount for each letter of the bases, the first letter in the bases, I could use AGTC, so that's 4 different letters I could use. And for the second letter, I could use AGTC. That's another 4 letters I could use. So I would actually only get 16 unique combos, which isn't sufficient to code for 40 unique amino acids. However, if I use 3 letters, meaning like the first letter of the sequence, the second letter of the sequence, and the third letter of the sequence, my first letter can be AGTC, my second letter can be AGTC, and my third letter can be AGTC, at least in the DNA. And in the RNA, it could be AGUC for each of those letters. But either way, if you do the math, 4 times 4 times 4, I can get 64 unique combinations in this sequence. And if I did 5 letters, it would actually be 4 times 4 times 4 times 4 times 4, which is 1,024 unique different combinations, which is too many. So generally speaking, we want to use the smallest number of letters to give us the just enough to cover the 20 unique amino acids I want to code for. So that's why, generally speaking, triplets is the best amount for combination, or the best amount of DNA bases we want to pair with each unique amino acid. Okay, so there are 20 possible amino acids that can be used, and there are 64 different codons that can be created from 3 letters. All right, this is a codon chart that's specific to mRNA, and all the codon charts we're going to give you is always for the mRNA. When you find out the sequence of something, whether it's DNA or whether it's tRNA or whether it's mRNA, you have to get it to the mRNA sequence for these charts to be useful. Okay? So don't just be using tRNA, don't just use your DNA and then assume that those letters are also the same on this chart. It doesn't work that way. You need to convert it to the mRNA before you can use these charts. But anyways, for those 3 letters that you have, 61 of those combinations will result in an amino acid, while 3 of them don't result in an amino acid at all. They result in a stop codon. And this stop codon is a sign that just tells your the ribosomes that's making the protein to go, hey, you're done, release the protein. We're done with this mRNA and final protein combination. All right. What that does mean, though, is because there's 20 amino acid, a lot of those codons, a lot of those triplet sequences code for the same amino acid. That's called redundancy, meaning more than one codon may specify a particular amino acid. So, for example, all of these code the same amino acid called threonine. All right. And just a heads up, all of these letters are the abbreviations of the actual name of the amino acid. On the later chart, you'll see the full name. All right, but no codon specifies more than one amino acid, meaning that UUA is always going to code for leucine. It's not going to suddenly code for phenylalanine. It's always leucine. So maybe more than one codon can lead to the same amino acid, but that codon always leads to that amino acid. It doesn't lead to more than one type of amino acid. All right, so this is actually a more specific codon chart, and again, it's still for mRNA. So when you're looking at a sequence and it's giving you DNA or tRNA, you need to convert it to mRNA before you can use this chart. But when you see ser, that's actually, the full name is serine. When you see pro, the full name is proline, glutamine, all that good stuff. Another amino acid codon I want to point out on this chart is AUG, which is going to become significant because it's always the first amino acid in a proper amino acid chain. Okay, so all amino acid chains start with AU gene, which gives you the amino acid methionine. And methionine is the amino acid that's needed to kickstart the ribosomal reading of mRNA. And these are alternative charts for mRNA. The way to read them, if you're not used to it, is let's say you have the sequence AGU on the mRNA. The way you read it is my A is here for my first base. My G is the second base is here. So that means I'm now looking at this box, which meets the first base A and the second base G. And then within that box, I'm looking for my third base, which is U. In this case, U is here. And that's how I know that this amino acid is serine, S-E-R. If you want to use this chart instead, you can then read it. The way this one reads is the first codon is the one most center and the last codon is the one to the outer edge. So you kind of read from the center on out. So in this case, if I want AGU, I start with A, then I go to G, then I go to U, and it leads me to serine. Both these charts are acceptable. Use the one that works the best for you, and you can use it as a reference. I'm not requiring you to memorize these charts, but you should be able to use these charts to help convert a codon sequence on the mRNA to determine which proper amino acid it will lead to. Okay, the other thing is because you're reading in triplets, the reading frame is very important, meaning your cell's ability to go, okay, this is the first triplet I'm going to use, so then the second triplet, the third triplet, the fourth triplet are this. It knows which 3 letters to use for each amino acid. If the reading's a little bit off, if you shift where the codon you're looking at is located, that can actually lead to the rest of the sequence being read incorrectly. And you'll see examples of that when we go over mutations, especially. Now, another thing to point out is this letter codon chart is universal to all life, meaning all cells and all living things use the same letter sequencing and translate it in the same way, meaning they lead to the same amino acid. So if you take the gene from a plant cell and you can somehow insert it into an animal cell, that animal cell will start producing the same protein from that gene that it took from the plant cell. Okay? So here we have an example of a tobacco plant expressing a firefly gene. And here you have an example of a pig expressing a jellyfish gene. And in fact, viruses take advantage of this because some viruses can insert their DNA to our cells and have our cells start expressing their viral genes and start making proteins that the virus needs to replicate, for example. And we'll learn more about that when we learn about viruses in the next unit, basically. Okay, so this is just a recap. You don't have to write all of these down again, but just reminding you, messenger RNA is the main one that holds the actual amino acid sequence code, while tRNA is going to be a helper RNA that's going to help bring and deliver my amino acid to the ribosome. And the ribosome is the actual location that the mRNA is going to be read and used to help build the amino acid sequence that's my protein. All right? The ribosome mainly consists of ribosomal RNA. And we're going to go specifically into tRNA and ribosomal RNA today. Okay? So kind of like a rough recap, the mRNA is sort of the message, the instructions. My tRNA is going to interpret those instructions by matching those instructions to each unique amino acid. And the machine that's basically reading and helping pair the tRNA with the mRNA and bind and make all the bonds between the amino acids, that's the ribosome, which contains our RNA and some other proteins that help this process out. Anyways, for tRNA, just like all RNA, it's made in the nucleus by RNA polymerase. And in this case for tRNA, each tRNA is specific to the amino acid. Now, most of the tRNA sequence is actually the same. So if you actually follow very carefully, this is a single strand of RNA, but then it's folded up really funky. But the last section of this tRNA is actually a location to string and connect an amino acid. And the amino acid will be unique to these 3-letter sequences right here, known as the anticodon. Okay? Generally speaking, when we ask you to give us a tRNA, we don't ask you in bio honors to tell us the entire sequence of the tRNA. We're asking you to give us a unique triplet sequence at the end. This triplet sequence on the anticodon is actually very important because it will determine what amino acid ends up attaching to the tRNA, and it will bring that amino acid to the mRNA that will correctly complement this tRNA sequence. So for example, an mRNA sequence of AUG will form hydrogen bonds, the proper hydrogen bonds that will allow it to connect to and complement the tRNA sequence UAC. So when the ribosome actually sees that tRNA sequence, it will correctly match it. If the mRNA sequence codon is up, it will correctly match the tRNA to it and then yank the amino acid that tRNA is carrying off the tRNA and add it to the protein that it's forming. Now, generally speaking though, basically the translation process occurs in 2 steps. I don't know why these quotation marks are there. But first, you have this plain empty tRNA with its anticodon, and then the amino acyl tRNA synthetase whole job is to find the correct amino acid and hook it onto the tRNA. All right? And then once that amino acid is hooked onto the tRNA is ready to be floating around. And when the ribosome realizes and notes that, hey, this tRNA matches my mRNA codon that I'm reading, it will bring that tRNA to the ribosome and pull off the amino acid that's at the end of the tRNA and use it. Okay? And then once that amino acid is taken off the end, the tRNA is empty again and can be reused by the amino acyl tRNA synthetase to attach a new amino acid. And then that tRNA can go back and be used again by a ribosome to bring the amino acid. Generally speaking, the first 2 letters of this anticodon must exactly match the mRNA, but this last letter isn't bound super tightly, so it's sometimes what we call a wobble, and actually accounts for the reason why a lot of the times, not always, but a lot of the times, the last letter of the codon in the mRNA doesn't have to be specific, meaning you have CG for the first 2 letters of the codon, and then the last letter of the mRNA codon could be anything, and you're always going to get arginine. All right? Or in this case, you might have for AC, the last letter doesn't really matter. You're always going to get threonine. For other ones, it does matter. For example, in these, a lot of these other ones, it can be one of two choices. But for the most part, it oftentimes that last letter isn't as hypercritical. Also, you might notice a lot of the times, if it is kind of the last letter is sort of important, it's usually of the same ring structure, meaning it could be U or C, both pyrimidines, or it could be A and G, both purines, double rings, to determine whether it's glutamic acid or aspartic acid. So just a heads up, that last wobble, because the last letter isn't as hypercritical as the first two letters, that often determines which codons kind of end up resulting in the same amino acid. Okay, so this is sort of a picture of the enzyme helping to add an amino acid to the end of the empty tRNAs. And then once they're released, they can then go to the ribosome to help release amino acids that are being used to build the protein. And then when it's empty again, just be recycled and have another amino acid attached to it. So the tRNAs don't disappear once they're used. Once they're emptied out, they get to go back and get reused. Okay, so now going to the ribosomes are the site of translation that basically form the peptide bonds between the amino acids. It's made of rRNA and proteins. So rRNA is an example of an RNA that acts a little bit like a protein. It can actually function a little bit like an enzyme. And its whole job is it helps hold onto and facilitate the formation of the bonds between the amino acids of the chain. All right? But it does it with proteins. So the combo of rRNA and proteins actually form the ribosome. So if you look at the ribosome, there's a smaller subunit and a larger subunit. All right. So what it actually does is between the large subunit and the smaller subunit, what they tend to do is they clamp on to an mRNA. And then their whole job is in their 3 sites within the ribosome, which we're going to go over today. Each of those sites is a location for a tRNA to pop in and bring an amino acid buddy to the party. And the whole job of the ribosome is to make sure that the correct tRNA comes in, and then when the correct tRNA comes in, it pulls off the appropriate amino acid and adds it to the growing chain. So that's the basic idea. Okay, for the ribosome, there's going to be 3 main sites that are really important, and it kind of runs opposite the direction of the way you see it in the ribosome. All right, so in the ribosome, the A site is the site that's used first. Then the P site is the site that's used second. And then the E site is the site that's used third. So it runs in the ape direction. So the first site is basically where a new amino acid on a tRNA comes in. And then the P site is where the main protein is held. And then once it kind of all shifts down to the E site, that's when the tRNA's protein portion, the amino acid has been popped off, and the tRNA is emptied out, and it can then be released. And then this is just going to be kind of what happens at each point the amino acid is added on. This is kind of confusing for me to explain on a 2D image. So what I'm going to do is I'm going to play a quick video to kind of show you what's happening after I kind of go over this sequence. So I'm going to first explain what's happening per sequence, which might be a little bit confusing. Then I'll play a video at the end just so that you kind of see what's happening. Okay, so initially what happens is this whole mRNA thing, the beginning of it might be something else. It might be the GTP cap and stuff like that. But what's actually happening is towards the front of the mRNA, you have a start codon, the AUG start codon. Okay? Wherever the start codon is, the tRNA that's holding methionine is going to clamp onto that start codon, and it's going to initiate a structure where the lower ribosomal unit is going to clamp on. That initial structure between the methionine tRNA, the mRNA, and the lower bottom part of the ribosome is going to induce the large upper part with the 3 sites, the A, P, E sites, to clamp on. So now we have this sandwich of my ribosome sliding down the mRNA, and the ribosome is going to slide down the mRNA in the 5' to 3' direction. It's going to move down the mRNA at each codon after the methionine start codon. Okay, so pretty much all proteins in a complete sequence always has methionine at the start. Sometimes our problems won't start with methionine, but that's not a true protein sequence. That is like an excerpt of the middle of the protein sequence, but not the full one. If we give you a full protein sequence, it'll begin with methionine. Okay, then elongation happens. So what happens is, as the ribosome sort of slides down the mRNA one codon at a time, a new tRNA with the amino acid that is next wanted in the sequence is going to hook onto the A site. At that point, when the tRNA with the amino acid is at the A site, the protein that's at the P site, that's the already so far built sequence at the P site, is going to basically shift its entire amino acid sequence and form a peptide bond onto the new tRNA. And then that whole tRNA at that A site is going to slide down to the P site. So it's going to move down to the P site, and that emptied out tRNA is going to move over to the E site. So this whole thing, again, the whole ribosome is shifting this way, and these guys are shifting down in that direction as the ribosome is moving from 5' to 3'. And then that emptied out tRNA leaves. Okay, so that's called translocation, when the whole mRNA gets slid down as the ribosome moves on to the next codon. And you kind of repeat this over and over as you go into each new added amino acid and you reach each new codon. Then when you hit the very end of the mRNA sequence, or maybe not the very end of the mRNA sequence, it might be somewhere in the middle if there's a mutation. But if you get later in the mRNA sequence and you hit a codon that translates to one of the 3 stop codons on the mRNA, that stop codon doesn't lead to a new tRNA coming into the A site. Instead, what it does is it brings in a release factor protein into the A site, and that release factor protein is going to induce hydrolysis that's keeping the ribosome structure together, and it's going to pop everything off and release the protein, release the ribosomal pieces, and release the mRNA. That mRNA can then be read by another ribosome, and these ribosomal pieces can then clamp onto another mRNA if it so wants to. Okay, so all of these pieces can be reused. The mRNA over time breaks down a little bit, so the mRNA can be used a few times, but maybe like a few hours later, it'll be a little bit broken down, and a day later for sure it'll be gone. Okay, so if you want more of that protein, you're going to need new mRNA to replace this busted copy. But it can be used multiple hundreds, if not thousands of times before it breaks down. So all these pieces can be reused a decent amount of times. And then after that protein is popped off, that protein, that amino acid here, it's a lot longer than what's shown in this cartoon, but then that's the primary structure protein, and it can then be folded up into eventually the more complex structures of the tertiary protein or quaternary protein, which are the protein folded structures that can do work. Okay, so this animation goes over the translation process. So what first happens is, this is my tRNA with its amino acid. It has to make a complex with the lower part of the ribosome and the start codon and the mRNA, which is down here on the green thing. Once it connects the start codon and tRNA and lower part of the ribosome, the top part of the ribosome can clamp on. At that point, you can start actually making the protein. So the A site is the location of incoming tRNA with the new amino acid, while the P site actually has the established running polypeptide sequence. And the E site is where all emptied out tRNA go to. So first, at the A site, tRNA come in with amino acids, but the tRNA is not an exact complement of the mRNA. It can't come in. So until a matching tRNA with the appropriate anticodon that matches the mRNA comes in, when that happens, such as this one, what's going to happen is the polypeptide on the P site is going to make a bond to the new incoming amino acid. The whole ribosome is then going to shift down the mRNA, and the incoming tRNA is now moved to the P site, and it has the growing amino acid chain. The empty tRNA at the E site is then going to be let go, and this tRNA can be recycled and used to add new amino acids. All incoming new amino acids are going to be added at each new codon until the stop codon is reached. And at this point, that's going to initiate the termination part, where a release factor comes into the A site and creates a little bit of hydrolysis that disconnects everything. And at that point, this polypeptide sequence is going to be released, so it can be folded up as a protein, and the remaining parts can be reused. The mRNA can be reused, and the ribosome. Okay, so now I'm going to go over some practice problems with DNA and mRNA and tRNA, and I hope that makes this make a little bit more sense. So let's say we have our 3' sequence of DNA of DNA, and this would be a template strand, and our mRNA is going to be 5' to 3' always. So in this case here, that's easy. The complement to a strand is always the opposite orientation. So if I get the complement to this DNA, it will be 5' to 3', which is the correct direction. And then my complement mRNA structure will be this. Generally speaking, when you look at your DNA, it's a good idea to kind of draw a line at every 3 bases. And then as you write your mRNA, you kind of want to add a little bit of a gap between the mRNA. This will prevent you from making mistakes later on when you actually read the codons. It's just a lot easier if you write them out in triplets. Then, when you're trying to figure out the amino acid sequence, you want to use the mRNA sequence. So even if you're asked to, on a worksheet, come up with the anticodons on the tRNA, you still ultimately want to use the mRNA for the amino acids. So the tRNA is, again, the complement of the mRNA. And if you notice, the complement of the mRNA is 3' to 5', because mRNA is always, always, always 5 to 3. It actually matches the DNA that's 3 to 5', with the exception of the fact that it's using U's instead of T's. But it complements the mRNA. So U to A, C to G, U to A, G to C, U to A, G, or A to U, G to C. But it's a complement. But when you're actually asking for the amino acid sequence, these codon charts that I have in the slides, they are always, always, always for the mRNA. So you're going to ignore the tRNA, and you're going to use the mRNA for this stuff. And then if you look for UCU here, UCU, that is serine. And then GAG, GAG over here matches to glutamine. And then that ends up being the amino acid sequence using the mRNA. I'm not too sure why these boxes are over here. Okay, so that's sort of how you read them. It is very important that you watch out for the codon chart, because if we give you 5' to 3' over here, your DNA, your mRNA sequence is not the complement to this, because the complement to this will end up being 3' to 5', which is not mRNA. mRNA is always 5' to 3'. So there's a few different approaches you can take here. One is you find the complementary DNA, the actual template DNA strand. And then you can take that template DNA strand, break it into codons, and convert that to an mRNA. Another thing you may notice, though, it's 5 to 3. So if you look at the DNA strand, 5 to 3 is basically the same, with the exception of the fact that you're using U's instead of T's for the mRNA. But the main idea is you just want the 5 to 3 sequence from the DNA. If the DNA is giving you the 5 to 3 sequence, you just kind of make it mRNA by just switching it to U's. But if you're using the 3 to 5, you have to complement it to get the 5 to 3 mRNA sequence. Then from there, again, same deal. You use the mRNA to make the amino acids. AGA leads to arginine. CUC leads to leucine. etc. And if you need the tRNA, you just get the complement of the mRNA. But do not use the tRNA to get the amino acids. That's not how it works. All right, I have a lot of videos. I think these videos are very helpful for kind of visualizing and thinking about transcription translation as a whole. And then over here, this kind of activity, if you run through it, also might help you understand gene expression a little bit more. It's a little bit more cartoony, less specific than what we've gone over, but it'll help you spot the A, P, and E sites. And then this is just kind of a summary of what we went over, if you want to look over that. And finally, this was the vocabulary we touched on. Bye.